Section 1. Dependence on plants. All animals and humans rely on plants for food. Photosynthesis. Green plants produce their own food through photosynthesis, making them autotrophs. Organisms depending on them are heterotrophs. Sunlight dependency. Plants use sunlight to synthesize organic compounds, forming the energy base for all life on Earth. Key roles of photosynthesis. Primary source of food, sustains all living forms. Oxygen production, vital for respiration. Focus, explores photosynthetic machinery and reactions converting light energy into chemical energy. Photosynthesis requirements, chlorophyll, light, and CO2 are essential for photosynthesis. Experiment 1. Tested starch formation in variegated leaves or leaves partially covered with black paper. Result, photosynthesis occurs only in green parts exposed to light. Experiment 2. Leaf partially enclosed in a test tube with KOH, absorbs CO2, while the other half exposed to air. Result, exposed part tested positive for starch, proving CO2 is required for photosynthesis. Joseph Priestley, 1770. Observed that a candle extinguishes and a mouse suffocates in a closed space. Adding a mint plant allowed the candle to burn and the mouse to survive. Conclusion: plants restore air by removing what animals and candles consume. Jan Ingenhousz, 1790s. Experimented with plants in sunlight versus darkness. In sunlight, green parts of plants released oxygen, seen as bubbles in aquatic plants. Conclusion: sunlight is essential for oxygen release and air purification by plants. Julius von Sachs, 1854. Showed that plants produce glucose during growth, storing it as starch. Identified chloroplasts as the site of glucose production in green parts of plants. T.W. Engelmann, 1843–1909. Split light into spectral components and illuminated green algae in a bacterial suspension. Found oxygen evolution occurs in blue and red light, defining the first action spectrum of photosynthesis. Key photosynthesis knowledge, 19th century. Plants use light, CO2, and water to make carbohydrates, example glucose. Empirical equation, CO2 plus H2O plus light rightward arrow CH2O plus O2. Cornelius van Niel, 1897–1985. Demonstrated photosynthesis as a light-dependent reaction, with hydrogen donors reducing CO2 to carbohydrates. Found O2 comes from water, not CO2, proven using radioisotopic techniques. Correct equation, 6CO2 plus 12H2O plus light rightward arrow C6H2O6 plus 6H2O plus 6O2. Note: photosynthesis is a multi-step process, not a single reaction. Section 2. Photosynthesis location. Occurs in chloroplasts within mesophyll cells of green leaves. Can also occur in other green parts of plants. Chloroplast structure. Contains grana, stacks of thylakoids, stroma lamellae, and stroma, matrix. Membrane system traps light and synthesizes ATP and NADP. Reactions in photosynthesis. Light reactions occur in the membrane system, directly driven by light. Dark reactions occur in the stroma, rely on ATP and NADP from light reactions. Key insight: dark reactions do not mean they occur in darkness but that they are independent of direct light. Types of pigments in photosynthesis. Leaf pigments. Plants contain multiple pigments contributing to their shades of green. Chlorophyll A, bright/blue-green. Chlorophyll B, yellow-green. Xanthophylls, yellow. Carotenoids, yellow to yellow-orange. Pigment functions. These pigments absorb light at specific wavelengths for photosynthesis. Chlorophyll A is the most abundant pigment and plays the central role. Absorption spectrum, figure 11.3a. Chlorophyll A absorbs maximally in the blue, 430 to 450 nanometers, and red, 640 to 680 nanometers, regions. Chlorophyll B and carotenoids extend the range of absorbed light. Action spectrum, figure 11.3b. The highest rate of photosynthesis corresponds to wavelengths where chlorophyll A absorption is strongest, blue and red. Key insight: there is no complete one-to-one overlap between the absorption spectrum and the action spectrum, figure 11.3c, as accessory pigments contribute to photosynthesis beyond chlorophyll A. Light reactions, photochemical phase, involve light absorption, water splitting, oxygen release, and the production of ATP and NADP. Light harvesting complexes, LHC. Pigments are organized into photosystem I, PSI, and photosystem II, PS2. LHC contains pigment molecules, antennae, that absorb light of different wavelengths. Reaction centers. Each photosystem has a unique chlorophyll A molecule as the reaction center. PSI, reaction center is P700, absorbs light at 700 nanometers. PS2, reaction center is P680, absorbs light at 680 nanometers. Process highlights. Light energy excites pigments, transferring energy to the reaction center. High-energy electrons are passed to the primary acceptor, initiating the electron transport chain. Figure 11.4 shows how photons excite pigments, transferring energy to the reaction center, leading to electron movement towards the primary acceptor. Electron transport. Photosystem II, PS2. Chlorophyll A in PS2 absorbs 680 nm red light, exciting electrons to a higher energy level. Excited electrons are transferred to an electron acceptor and then passed through an electron transport system, cytochromes. Photosystem I, PSI. Electrons from PS2 are transferred to PSI, where they are re-energized by 700 nm red light. These electrons reduce NADP to form NADP plus H, an energy-rich molecule. Z-scheme describes the flow of electrons. Starts in PS2 rightward arrow electron transport chain rightward arrow PSI rightward arrow NADP. Named for its characteristic Z shape showing uphill and downhill energy movement. Key concepts. Downhill movement, electrons move through cytochromes via redox reactions. ATP and NADP are generated during the process to power the Calvin cycle. Figure 11.5 illustrates the Z scheme. PS2, excitation rightward arrow electron transport. PSI, re-excitation rightward arrow NADP reduction. Section 3. Splitting of water and photophosphorylation. 1.6.1 Splitting of water. Purpose, supplies electrons to photosystem II, PS2. Process. Water, H2O, is split into protons, H. Oxygen, O2. Electrons, E. Equation. 2H2O rightward arrow 4H plus O2 plus 4E. Location, occurs in the lumen of the thylakoid membrane. Significance, O2 is released as a byproduct, and protons contribute to the proton gradient used for ATP synthesis. 1.6.2 Cyclic and non-cyclic photophosphorylation. Photophosphorylation, ATP is synthesized from ADP and inorganic phosphate in the presence of light. Types. Non-cyclic photophosphorylation involves PS2 rightward arrow PSI via the Z scheme. Produces ATP, NADP, and releases O2. Cyclic photophosphorylation. Only PSI is involved, electrons cycle back to PSI. Produces ATP only, no NADP or O2. Occurs in the stromal lamellae, lacking PS2 and NADP reductase. Significance. Non-cyclic produces energy and reducing power for the Calvin cycle. Cyclic ensures additional ATP when required. Figure highlights. Figure 11.5, Z scheme showing the flow of electrons in non-cyclic photophosphorylation. Figure 11.6, cyclic photophosphorylation pathway illustrating the electron cycling within psi. Section 4. Chemiosmotic hypothesis. ATP synthesis in chloroplasts. ATP synthesis is explained by the chemiosmotic hypothesis. A proton gradient across the thylakoid membrane drives ATP formation. Proton gradient formation. Splitting of water produces protons, which accumulate in the lumen of the thylakoid. Electron transport, protons are transported across the membrane by carriers, reducing protons in the stroma and increasing them in the lumen. NADP reduction, protons are used to reduce NADP to NADP on the stroma side, further depleting stroma protons. Importance of the proton gradient. Gradient breakdown, protons diffuse back into the stroma through the CF channel of ATP synthase. The energy released drives conformational changes in the CF particle, catalyzing ATP formation. ATP synthase structure. CF, embedded in the thylakoid membrane, facilitates proton diffusion. CF, protrudes into the stroma and synthesizes ATP. Chemiosmosis requirements. Requires a membrane, proton pump, proton gradient, and ATP synthase. Energy from the gradient is used to form ATP. Products of chemiosmosis. ATP and NADP produced are immediately used in the Calvin cycle for CO2 fixation and sugar synthesis. Calvin cycle and biosynthetic phase. ATP and NADP usage. Light reaction products. ATP and NADP are used in the biosynthetic phase to fix CO2 and synthesize sugars. Verification, biosynthetic processes continue briefly after light is unavailable, confirming dependence on light reaction products. CO2 fixation pathways. C3 pathway. First product, 3-phosphoglyceric acid, PGA, 3-carbon compound. Found in most plants. C4 pathway. First product, oxaloastatic acid, OAA, 4-carbon compound. Found in plants adapted to hot climates. Primary acceptor of CO2. RUBP, ribulose bisphosphate. A 5-carbon ketose sugar. Accepts CO2 to form PGA. Discovered after years of experimentation. Calvin cycle, C3 cycle. Stages. Carboxylation. CO2 is fixed by RUBP, catalyzed by RU-BisCO, to form 2 molecules of PGA. Reduction. PGA is converted into glucose using 2 ATP and 2 NADP per CO2 molecule. Regeneration. RUBP is regenerated using 1 ATP per CO2 molecule. Energy requirement. For 1 glucose molecule, 6 CO2 molecules. 18 ATP and 12 NADP are needed. Key insights. Calvin cycle occurs in all photosynthetic plants, irrespective of their CO2 fixation pathway, C3 or C4. Cyclic photophosphorylation helps balance the ATP and NADP ratio needed for the Calvin cycle. The C4 pathway. C4 plants adaptation. Found in dry tropical regions, C4 plants use the C4 pathway to fix CO2. First product, oxaloastatic acid, OAA, a 4-carbon compound. Efficient under high light intensity and temperature with high productivity. Krantz anatomy. Bundle sheath cells surround vascular bundles in C4 plants. Characteristics. Rich in chloroplasts, no intercellular spaces. Impermeable to gas exchange. Mesophyll cells and bundle sheath cells work together. Hatch and Slack pathway. CO2 fixation. Primary acceptor, phosphenol pyruvate, PEP, in mesophyll cells. Enzyme, PEP carboxylase, PEPcase, forms OAA. Transport. OAA converts to other 4-carbon acids, example malic acid, transported to bundle sheath cells. Decarboxylation. CO2 released in bundle sheath cells for the Calvin cycle. Regeneration. 3-carbon molecule returns to mesophyll and regenerates PEP. Comparison with C3 plants. C3 plants, Calvin cycle occurs entirely in mesophyll cells. C4 plants, Calvin cycle occurs only in bundle sheath cells. Key enzymes. C4 plants lack RU-bis-CO in mesophyll cells but use PEPcase. Bundle sheath cells in C4 plants have RU-bis-CO for the Calvin cycle. Section 4. Definition. Photorespiration occurs when RU-bis-CO, instead of fixing CO2, binds with O2. This results in decreased CO2 fixation efficiency. Mechanism. Normal reaction. RUBP plus CO2 rightward arrow 2 molecules of 3-PGA, Calvin cycle. In photorespiration. RUBP plus O2 rightward arrow phosphoglycolate, 2 carbon, plus 3-PGA. Consequences of photorespiration in C3 plants. No ATP or NADP is produced. CO2 is released, and energy is wasted. Results in reduced photosynthetic efficiency. Rubisco's role. RU-bis-CO can bind to both CO2 and O2 due to its dual affinity. The binding depends on the relative concentration of CO2 and O2. C4 plants and photorespiration. Photorespiration is minimized due to Krantz anatomy. CO2 is concentrated in the bundle sheath cells. This reduces Rubisco's interaction with O2. Importance. Photorespiration limits productivity in C3 plants, especially under high temperatures. C4 plants, with their efficient CO2 concentration mechanism, show better adaptability to such conditions. Roti-Curry. Each letter stands for a key concept. R, RU-bis-CO. Enzyme with dual affinity for CO2 and O2. O, oxygenation. RU-bis-CO binds to O2 instead of CO2 during photorespiration. T, 2-carbon. Produces phosphoglycolate, 2-carbon compound, instead of 3-PGA. I, inefficiency. No ATP or NADP is produced, energy is wasted. C, C4 plants. Concentrate CO2 in bundle sheath cells to minimize photorespiration. U, utilization of CO2. CO2 is better utilized in C4 plants, increasing efficiency. R, release of CO2. Photorespiration leads to CO2 release in C3 plants. R, reduction in yield. C3 plants suffer productivity losses in high temperatures. Y, yield advantage. C4 plants thrive in hot conditions due to reduced photorespiration. Factors affecting photosynthesis. General factors. Internal factors. Number, size, and age of leaves. Chloroplast count and internal CO2 concentration. Chlorophyll content, influenced by genetics and growth. External factors. Light, temperature, CO2 concentration, and water availability. Law of limiting factors, Blackman. The rate of photosynthesis is limited by the factor closest to its minimal value. Light. Influenced by quality, intensity, and duration. Light saturation occurs at 10% of full sunlight. Beyond this, excess light can degrade chlorophyll, reducing photosynthesis. CO2 concentration. CO2 is a major limiting factor, atmospheric levels, 0.03 to 0.04%. C3 plants, saturation at greater than 450 ll superscript 1. C4 plants, saturation at 360 ll superscript 1. CO2 enrichment improves productivity in greenhouse crops like tomatoes and bell peppers. Temperature. Dark reactions are enzymatic and temperature sensitive. C4 plants, higher temperature optima than C3 plants. Temperature preference varies based on habitat, tropical versus temperate. Water. Indirectly affects photosynthesis via stomatal closure, reducing CO2 availability. Water stress causes leaf wilting, reducing metabolic activity and surface area. Summary of photosynthesis. Light reaction. Occurs in thylakoid membranes. Produces ATP, NADP, and releases O2. Driven by PSI, P700, and PS2, P680. Carbon fixation. C3 pathway, RU bis CO fixes CO2 to form PGA using ATP and NADP. C4 pathway, CO2 is first fixed into a 4-carbon compound in mesophyll cells, then processed in bundle sheath cells via the Calvin cycle. Photorespiration. Wastes energy in C3 plants under low CO2/high O2 conditions. C4 plants avoid photorespiration by concentrating CO2 in bundle sheath cells. Batsman. Each letter corresponds to a key concept. B. Blackman's law. The law of limiting factors determines the rate of photosynthesis based on the factor nearest to its minimal value. A. Available light. Light intensity, quality, and duration affect photosynthesis. Saturation occurs at low levels, while excess light can degrade chlorophyll. T. Temperature. Dark reactions are temperature sensitive. C4 plants, higher temperature tolerance. C3 plants, lower temperature optimum. S. Stomatal closure, water stress. Water affects photosynthesis indirectly by closing stomata, reducing CO2 intake, and causing leaves to wilt. M. Mesophyll cells, C4 pathway. Initial CO2 fixation in mesophyll cells forms a 4-carbon compound before the Calvin cycle in bundle sheath cells. A. Atmospheric CO2. CO2 is a limiting factor. C3 plants respond to higher CO2 levels, greater than 450 ll superscript 1. C4 plants, saturation occurs at 360 ll superscript 1. N. NADP and ATP usage. Products of light reactions drive the Calvin cycle to synthesize sugars.